yes, it probably was a little more, because, energetically the most stable way of organizing three-dimensional space could take other forms. But that's what I think, but I haven't seen the first cell, so I don't know. How did the very first membrane appear? That is a very interesting question, because that's sort of a chicken-egg question, right? And that may have been a very long process, because membranes initially could probably partition into the bilayer spontaneously. But this was a very laborious process, because you had to construct the protein so that it could partition into the bilayer. And so then later on you wanted to make sure that the partitioning is somehow controlled, and that you need only small sequence elements for doing that. And that's how these machines develop. Now I could go on and speculate a long time, but I think it's inappropriate in the short time to go into these speculations. But it's easily conceivable how such things could have occurred, because we don't know in detail. Now let me see, how did the very answer this, answer this, answer this, answer this. In your opinion, what is the state of university education and quality in the USA nowadays? How do you like Russian university education? Well, the university education in the United States is, of course, excellent. And the United States has always educated a small elite, but was not very good in educating the masses. And so therefore there are, in my opinion, deficiencies in educating the broad population. They have really spent a lot of time to educating an elite, and it has been recognized, and therefore a lot of money is being spent to improve elementary education. This is why people can still persuade people that evolution doesn't exist. Right? This is much more difficult in Russia, because in Russia people have a much better basic education. And of course they have magnificent university education as well. But you had a lot of problems. You had wars, you had all sorts of upheavals, you had the magnificent tradition of Russian science was cut down several times. I come from a country, Germany, where this happened the same way. And science is like a mimosa. If you mistreat it, if the mimosa gets out into the cold, it dies. And it takes very, very difficult to resurrect, to nourish science again. So science is like a very delicate plant which you have to nourish every day to really do all your efforts to make it happen. And this is a big problem. And of course, in the United States, they have not had a war. They have not had major upheavals, and so their university system really also benefited from the collapse of the European university system. All the Jews that were forced to emigrate from Germany all went to the United States. Many Russians that emigrated from Russia through all of the upheavals of Russia are also found in the United States. Many of them. Some of them, because many of them stayed here. And so, now I'm very optimistic for the future of Russia. And I hope that you, the next generation, will have the opportunity to bring back Russian science to the glory that it should have. And that it should be a major contributor to our scientific knowledge. And it will take a while. We're discussing private money, we're discussing all sorts of things. It is a huge challenge, but I'm confident that if there is no future war, no future upheavals, which I hope there will not be, at least in Europe and Russia and the United States and so on, that Russia will be back as it was in the past. It will be a great contributor to science. Which it already is, but could even be more. Dear professor, if you look at cell, where chemistry finished and biology starts. A very charming question. Actually, you cannot separate biology from chemistry. Biology is physics, is chemistry, is mathematics, is every one of those things. Right? Biology is not just looking to the microscope and see a worm moving. That is part of biology too. But biology is chemistry. I had a long discussion with Dr. Spulacek about chemistry in mitochondria, radical production of mitochondria, and so on and so on. So there is a tremendous amount of chemistry, and you cannot separate it.
How to think? Viruses are simply machines, molecular machines, or organisms. Living.
Viruses. Well, viruses are really not cells because they are really nucleic acid containing information modules, if you wish, that can enter the cells and that use the cellular machinery to replicate themselves. And that's what they have learned how to do. Well, I shouldn't say learn, and evolution just allowed this to occur. Some people say there are escape DNA bits and pieces from the cell that then have the ability to protect cells and to replicate.
Is there life in viruses? Are they heavy?
Is there life? In what? No. The viruses, if they don't have cells, don't live. You can store them in your tube, but they cannot replicate without cells. Of course, now we have learned to do it in vitro. We can take poliovirus RNA, we can take the enzyme, and we can make more poliovirus RNA. Right? So we have learned all these things, but the virus by itself needs the machinery of the cell. It's a parasite, so to speak. It's a parasite which lives from the machinery of the cell. Okay. Are there any evidences that human stem cells can be manipulated in such specific fashion as that of mice or rats? The question is, can you put the way I understand it, to what extent do human stem cells and mice stem cells differ? Well, of course they will differ to a certain extent, but many of the basic aspects of stem cell biology and differentiation can be worked out with mice. And of course, ultimately, you will have to repeat it in humans, with human stem cells. But much of the basic biology, how many stations do you have to go from an embryonic stem cell to a hematopoietic stem cell, to progenitor stem cell for white and red blood cells, and so on and so on. There are many stages of stem cell differentiation, and that has to be studied, of course, individually in man. But you can start beginning to study this in mice, because there are many advantages of doing this in mice, because it's easier to confirm your observations, because you can put the cell back into mice, but you will never be able to do this with humans, obviously. You know, before you can use stem cells in humans, it will take a very long time, because you want to make sure that the stem cell that you give into humans doesn't become a cancer cell, for instance. Right, so you have to know very much about the stem cells before you use them in therapy. And this will take a long time. Oh, the other question I didn't answer, what excites you in biology? Well, I spent 1 hour to get you excited. Lots of things, everything. You have to Einstein said once a wonderful thing. He said, I wasn't particularly intelligent, but I was just passionately curious. For those of you who speak German, ich war nicht besonders intelligent, aber leidenschaftlich neugierig. So that's what, and this passionate curiosity is something which a child has. A child wants to know everything. And this is what you have to retain in yourself, this curiosity of a child. And then everything will excite you. And in biology, some more, some less. But that's what is really, for me, the most exciting thing in biology. Of course, this happens in physics, it happens in chemistry. I'm not saying that it's limited to biology. Does there exist a measure of biochemical and biophysical differences in functioning membranes in the fixed type of cells? Mechanisms. Fixed type of cells, do you mean cells that have been fixed by aldehyde to look in the microscope? Or you mean a particular cell type? It is difficult for me to really decipher the question, but I offer you, when we have finished, that you come down and you talk to me privately, and then I will try to explain the question.
I have two questions in Russian, just translate. What was your first idea when you learned that you are a Nobel Prize winner?
Oh.
Not obligatory to answer.
Well, I mean, I was very overjoyed. I'm not sure whether I had a lot of ideas. The joy in science, the biggest joy in science comes every day when you find something or when you hear something new. The Nobel Prize is just, a sort of, yes, we like what you did and we give you this prize. I mean, there are many people who have done wonderful things, and it's a wonderful recognition, and it gives you a platform to do wonderful things. For instance, the work that I've done in Dresden or whatever. But I don't think about the Nobel Prize anymore. It has a short half-life. You think about it in the first couple of hours or days, and then you think about other things. Oh, more questions about Nobel Prize? In the fantastic film Fifth Element, there's an extraterrestrial body with much more than double DNA spirals. In your opinion, is it possible in nature? Well, you don't have to be an extraterrestrial body. The humble lily, the plant, has much more DNA than we have. But it's not information DNA that codes for particularly exciting and interesting things. So, yes, there could be organisms that have twice the DNA, but they may not be more coding for more intelligent, for proteins that give us the intelligence that we have. What do you mind about ideas of Albrecht Bühler about cellular mind? Well, I know Albrecht Bühler, and he's a very fervent scientist, and he thinks that the centriole is the center of the cell that has some sort of cellular intelligence. But he doesn't mean intelligence like the brain intelligence. It just orients the cells through microtubules and through other things, where it is, whether it should divide, whether not. And I think the term mind, in my modest opinion, is not a good analogy. I know Albrecht very well, Albrecht Bühler very well, but I think, that he has done beautiful things, but the nomenclature of mind I don't agree with. Professor, can you say 2 words about the global interaction between the cells inside human organism, about global cell community. Global cell community. You mean the interactions of all cells? Pardon?
The sort of globalism.
Well, I thought global issues are only in the G8 meetings discussed. No, I showed you one of the interactions of the global cell interactions when a bacteria is chased by a macrophage. Here you have a cell, bacteria, that is a very early kind of cell, derives similar to an early cell, and is chased by a cell that has a very long, the bacterial cell has a very long history of evolution, the same long history of evolution as ours, as a macrophage, but has much less DNA and has much less information. And so therefore, I have shown you the interaction that can take place between bacteria and eukaryotic cells. And this is a very, if you mean that by global cell community, if you mean it by that. There's also interaction between our cells and fungi, and many fungi infections. So there are quite a few interactions that can be studied. Okay, that's it. Do you think that DNA and cell formation is possible now anywhere under the seed or in the earth's crust from inorganic substances such as nitrogen, phosphate, and methane? No. As I said, the cell arose 4 billion years ago, and it probably, if it had arisen again, the other cells were so advanced that they probably have eaten it. Very much like if you look in human evolution. In human evolution, there was a very small founding family in Africa of about 1,000, 10,000 people who lived in Africa that left Africa more than 100,000 years ago. And all people on this planet, all people, whether it's the Eskimos or whether it's the blacks in Africa or whether it's the original people of Australia, all derived from that 100,000 people, from these 10,000 people that emigrated from Africa throughout the world. Now there were earlier emigrations from Africa, 600,000 years ago, even earlier. For instance, the Neanderthals were emigrants earlier from Africa, but they did not mix with the later emigrants from Africa that emigrated from Africa more than 100,000 years ago. So this is really sort of an analogy to what you were asking. But I wanted to make one little comment about this 100,000 years and the people from Africa, and that everybody in this world goes back, every human being goes back to this small population. Now 100,000 years, if you calculate 5 generations per 100 years, 5 generations, so it would take only 5,000 people, if one person represents one generation, it would only present 5,000 people, each representing one generation, to go back to the founding family in Africa. 5,000 people. That would be the population of a small city. And I think most of us don't think about it. Racial hatred, discrimination, all of these things are, from a biological point, utterly foolish enough, because we are all members of the same family. We are all cousins. We all come from the same small founding family in Africa. And it's only 5,000 generations ago. And so I think, it would be very important that this is being taught in schools So that racism, somebody who has a different skin or has a different something else in a human being is not being discriminated against, because we don't teach it in the schools. We don't teach it to school children. And they should know about it. I think it's a very, very important thing. But anyway, next question. Is the problem of cloning acute nowadays? What are the latest results of solving the problem of human genes? Now, the cloning, I don't know what you mean by cloning. A cloning of a person. Let's suppose I take a cell from you and or a nucleus from you, which has all your information, and put it into an oversite, an egg cell, take out the egg, and then the genetic program, excluding those that are contributed by mitochondria, will be from you, and that would be, if that could be possible, that would be a clone of yours, and this has been done in animals. But of course in humans it will not be done, because in animals we know when this is done, the animals are not normal. Dolly, you've heard, died much sooner than a normal cousin of Dolly. And so there are many, many problems that we haven't grasped yet. And one of them, of course, one of the reasons if you do nuclear transfer is that you don't go through meiosis, you don't go through the haploid phase. And in the haploid phase of there are many, many modifications, methylations, and other things, imprinting of the genome that you don't reproduce in when you do nuclear transfer. I don't want to go and discuss it in detail, but cloning of humans is not possible and hopefully will never be done. Incidentally, even if you would succeed, you would never get a complete copy because as you know, there is an environment which has shaped you significantly. It's not only your genes. What are the latest results of solving the problem of the human gene? I don't know what you mean by that. You mean the human genome? Human genome is very big. You may have read that Jim Watson, the discoverer of the string together of the structure of DNA, had recently his genome sequenced and published. So we know this sequence of the genome of Jim Watson, and sequencing becomes cheaper and cheaper, so that we will soon have individual genomes. So if you're an oligarch, you can afford to have your genome sequenced. It costs only $1 million. If you are, probably in 5 years it will cost $100,000, and then it will cost only $10,000. So if you're very interested in having your genome sequenced, but then will be all of these information people will decode what you may have in your genome. They may tell you you have a high chance for breast cancer, for prostate cancer, for brain cancer, or God knows what, or to become risky for Alzheimer's or whatever. So I think, you know, I'm not sure whether this is the way to go, but certainly for research and science it's very good. Now here, as you understand, many exciting, beautiful aspects of biology. However, every time you highlight a specific problem, you add it as a side note that our generation will probably not be able to solve it. When I said this, two reasons. First of all, there are many problems in biology that need to be solved. And even if you are old like me, and the next generation comes along, more problems will be made to be solved, because biology is extremely complex. Life is extremely complex. And so that we will not run out of problems. There's one philosopher who said once, human knowledge will be imperfect forever, but is indefinitely perfectible. And so we will never know everything. We can speculate, we can learn more, we can perfect our knowledge more. but we will not know everything. That doesn't mean that I'm implying a God or religion or anything like this. I mean, I'm not implying this. It's just saying that our knowledge will not be perfect because our brain is not perfect. We transform things in our brain that is very subjective, made for us to survive and not necessarily to interpret all the phenomena of nature in a correct way. So, but these are philosophical discussions we can have some other time. What problems then do you believe may be solved in our lifetimes? Plenty of them. Can we manage speed of cellular processes, gross DNA replication, and so on? From what depends speeds of cellular processes? Again, to make it short, all the kinetics in the cells are very, very carefully regulated. If you replicate DNA too fast, you make more errors. If you replicate it too slowly, it's not compatible with life. So the kinetics have been very carefully selected in evolution and are adjusted and optimized for life. Same thing happens in proteins. You can make proteins in a very slow fashion, but then they will have no errors in their amino acid sequence. But if you do it in a very slow fashion, it will not be enough to really keep up with the demand for proteins in the cell. From what depends speed of cellular process? Well, that's a very complicated question. You can look textbooks in biochemistry and they will tell you a little bit about it.
If no more questions, thank you very much. Mr. Blobel, absolutely brilliant presentation. Position, I'd like to give you this global.
перечитать. Это мне кажется, следует, что человек прошел не друзья на бактерии, они меня не понимают. Я тогда
después del paseo. Con tu cámara sale, pero... ¿Has visto? Esta putalía no tiene idea de nada. Sí, tú no de nada y puedes subir.
A ver, te lleva ya la mía. ¿Estás comiendo chicle? Vaya, está pillado. Dame uno. Te has hecho mi espalda. Te has hecho mi espalda. Mi espalda, ahí te vas a sacar. Ahí sale.
Ya, pero espérate, montar el compro que vengan aquí por la tarde. Es la tarde, no queréis. Ya, porque si voy a ir por la noche, si voy a ir por la tarde. Pero si la chica ha dicho que por la noche, ¿no?